introduction, and particularly to Professor Sen for inviting me. I came here via American Airlines, which was probably a mistake because my luggage is still in New York. I should have chosen Swiss Airlines, and then I would have had a tie on. Actually, I'm not wearing a tie in honor of Chuck Peru, who never wears a tie, and I see he's actually wearing one today, so now I'm totally confused because I thought genomics people didn't wear ties. So I'm going to talk to you about perhaps try and summarize for you some data on mutation analysis of breast cancer. And I've skewed this talk towards luminal type breast cancer because I thought Will Fawkes did a fantastic job of triple negative disease earlier on this afternoon. Here are my disclosures. So as Martine pointed out, it's been a bonanza of breast cancer genome sequencing studies over the last several years. In 2009 and 2010, you could get a Nature paper by sequencing one tumor, and by 2012, to get into Nature, you had to sequence hundreds of tumors. And the sort of pinnacle achievement of these sequencing efforts was the TCGA paper in Nature, which sequenced 507 breast cancer cases. This is a hard slide to read, and I only point this, not going to ask you to sort of memorize a long list of genes here, but I want to make one point. Look at this long list of genes here. These are the significantly mutated genes in breast cancer. These develop recurrent mutations in the disease. And look how long the list is of significance in ER-positive, HER2-negative disease, and how short the list is in HER2-positive disease, and in fact, strikingly short in triple-negative disease. That's because triple-negative disease is incredibly heterogeneous, and these significance tests don't seem to pick out very many genes. But in ER-positive disease, it picks up an extraordinary list of complexity and fascination that I just want to try and summarize for you. The study that we did was perhaps different from some of the studies done by the Genome Sequencing Consortia because we basically started our genome sequencing question with a hypothesis. And we focused on clinical trial samples and not samples of convenience from tumor banks. In this particular study design, we gave neoadjuvant endocrine therapy with an aromatase inhibitor, and which aromatase inhibitor we used, it turned out not to matter, but there was clear clinical benefit for the patient in that the aromatase inhibitors were inducing tumor regression and promoting breast-conserving surgery. But of course, in the course of this trial, we also got patient-by-patient information on endocrine responsiveness in the form of tumor regressions and changes in the proliferation index. And we could parse that information against the individual genomes to discover genes that were dominant with respect to determining the endocrine phenotype. And I just want to point out, I did this work with the American College of Surgeons Oncology Group, and Kelly Hunt was the chair of the breast committee. Nice picture of her there. So this is the flow diagram that we used in this experiment. We had frozen cores from the patients at baseline before therapy, and then basically we did a whole genome sequencing, and to extend the discovery exercise, in some patients we also did exome sequencing, and then we were able to compare patients' genomes based on whether the proliferation index was not fully suppressed by endocrine therapy or was suppressed by endocrine therapy to discover genes that were directing, if you like, estrogen-independent proliferation. This study involved an enormous number of people. It sort of takes a village, if you like, to do this kind of genome sequencing analysis. And I'd just like to point out Li Ding and Elaine Mardis, my close collaborators at the Genome Institute at WashU in the conduct of this study. The paper's published, and therefore I don't know I need to go into huge details into it in a short talk like this, but I was fortunate enough to look at some of the sort of deeper interpretation of the mutational spectra with two colleagues, Theodore Goldstein and Josh Stewart from the University of California, Santa Cruz. I think this gentleman's an interesting person to point out. He actually wrote the software for the iPhone, and when he got bored, he decided to become a bioinformaticist. And he generated this way of depicting the relationship between mutations and clinical features, which I think is rather interesting. You can obviously go to the original papers to see how this was constructed. There's about a 4,000 gene network analysis behind this RISTIC. But basically what it shows you at the bottom is luminal A breast cancer, obviously the better prognosis disease, and luminal B breast cancer at the top here. And you can see there's clinical associations with being luminal B, like high proliferation index and high grade. And then the various mutations, which are binary, are entered into this informatic program and allows you to see whether there's association with good or poor outcome features. And no surprise here that P53 strongly associates with poor outcome luminal B disease, not a surprise there. But interestingly, a new finding here that there's a gene called MAP3K1, which is a stress kinase. These are loss of function mutations, and that strongly associates with luminal A type breast cancer or indolent tumor features. It's like the gene and pathway for generating low-grade breast cancer. It's almost as if these breast cancers actually evolve not by increasing the proliferation rate, but perhaps by decreasing the death rate. Well, there's a sort of very novel biology on here which is currently undergoing exploration. So, for example, this gene here, MALAT1, is actually not encoding a protein at all. This is a long non-coding RNA with clustered mutations associating with poor outcome breast cancer. So, of course, that could open up an entirely new field of investigation. Now, I was fortunate to have this gentleman, Brian Brantine, working in the neighboring laboratory. Brian worked with me to look at every single mutation that we found to try and generate models for how these mutations were working. This is the summary we produced in the paper, but it's a rather complicated summary, which I tried to simplify for the sake of conversations with people who do clinical trial design. So green here are essentially gain-of-function mutations of various types. You can obviously gain gene function by gene amplification or with mutations that increase, for example, kinase functions, and this would be the case here in PIK3CA and AKT, for example. But also, of course, all-important loss-of-function mutations, and we discussed P10 a little bit earlier. But remarkably, sort of a relatively new finding is that ER-positive breast cancer has losses in this MAP kinase, the stress kinase pathway that feeds through to cell death. So this is basically making the cells survive in the stressful environment of an evolving tumor. And then, of course, there are various negative regulators down here, of course, P53 and RB, which basically negatively regulate E2F. And then these are all regulated by kinases, for example, these CDK4s, and I'll talk about those in a minute. And there's even negative regulators of negative regulators. It sounds complicated, I guess it is, but I'm going to talk a little bit as well about MDM2, which is a negative regulator of P53. So first of all, let's just talk about gain-of-function mutations in receptor tyrosine kinases. So interestingly, on the significantly mutated gene list, you won't find HER2 somatic mutation. And the reason for that is HER2 somatic mutation is relatively uncommon, occurs in about 2% of breast cancer overall, and can occur in ER-negative and ER-positive disease. But interestingly, most of patients with HER2 somatic mutations have HER2 normal disease. So these patients wouldn't normally get a HER2-directed therapy, yet they have a HER2 mutation. And as can be seen from this lollipop diagram, these HER2 mutations are clustering together, indicating gain-of-function. So there's gain-of-function mutations here in the kinase domain, and there's gain-of-function mutations here in the dimerization interface. You can say, well, 2% doesn't matter very much. That's a very rare population. But remember, the denominator of breast cancer is huge. So a 2% population produces about 4,000 patients a year in the United States alone, and that's about equivalent to the chronic myeloid leukemia population, which is routinely given HER2 kinase inhibitors. So if you think that 2% of breast cancer is unimportant, you also think that CML is unimportant, and I think that probably most people think you were wrong. Now this slide actually makes the point that these HER2 mutations are expressed, and also you can see here most of them have normal HER2 gene copy. Here you can see the mutant allele tends to be heterozygous, in keeping with the fact these are heterozygous gain-of-function mutations. These mutations are being studied intensely at this point. You can see that these kinase domain mutations can occur in the N lobe or in the C lobe around the ATP binding pocket. And you might imagine that the functional consequences of where these mutations are could be an important consideration, and that turns out to be the case, and I'll show you that in the next slide. These dimerization mutations in the extracellular domain are very precise. They only occur at S3110 or G309, and presumably they affect the dynamics of heterodimerization and cause activation, but we don't really completely understand that mechanism yet. The importance of where these mutations are is, I think, best illustrated by this mutation, L755S, because L755S is completely resistant to lapatinib, but sensitive to neratinib. Some tumors are equally sensitive to neratinib and lapatinib, but clearly a kinase inhibitor choice seems to be dictated by which mutation is present. And so as a result, in our trial where we're sequencing patients to find HER2 mutations, we're going to do the study with neratinib. And here you can see the trial design here, and we're interested in opening as many sites as possible because, of course, we have to find this 2% population with metastatic breast cancer. It's possible, and we don't know this yet, that metastatic breast cancer may be enriched for HER2 mutation. This is just a hint at this point. But interestingly, after we published the result, patients with HER2 mutation sort of came dropping out of the sky because patients are accessing various investigational genome sequencing programs, such as Foundation Medicine and various institutional efforts like the one at the Princess Margaret. And so at this point, we know of 5 patients with metastatic disease and mutations. But so far we haven't treated anyone with neratinib yet, and the reason for that is that patients are going to these mutation profiling when towards the end of their clinical history, when they've been treated with multiple other agents and the doctor doesn't know what to give them. And so many of them actually have brain mets, and many of them have a poor performance status, and so that just argues for getting the mutational profiles much early on in the course of the metastatic breast cancer if the trial is going to be successful. So that's, of course, a very interesting story. Another story we've heard a lot about today is this PI3 kinase story, and I'm not going to spend too much time on that. But, of course, we have this beautiful proof of principle study showing that, in general, in endocrine-resistant disease, targeting the PI3 kinase inhibitor, in this case with an anti-tumor antibiotic called everolimus, is effective in extending progression-free survival. But of course, rapamycin analogs are natural products. They have off-target effects. They're not the easiest drugs to use, although they do appear effective. And so it would be nice to use more precise agents, and Dr. Baselga gave you a beautiful list of these. We've just been studying one, which is called MK2206. This is an AKT inhibitor. And with Cesar Sanchez here, who is an ASCO Life Fellow in my lab, you can see here that under estrogen-deprived conditions, addition of MK2206 induces apoptosis in breast cancer cell lines that bear PI3 kinase mutations, or to Kent's point, are PTEN null. So these would be good selection criteria for treatment with this drug in the presence of estrogen deprivation. So we've gone on to start a program doing this in the neoadjuvant setting because it turns out that MK2206 is a relatively safe drug. And basically, we decided to focus on the PIK3CA mutant population because if we didn't see dramatic efficacy there, we were unlikely to see it elsewhere. So in this neoadjuvant study, they start an anastrozole without the second drug, fully establishing estrogen deprivation over 28 days. And during that time, we sequence the tumor, and if a PIK3CA mutation is present, we give them the AKT inhibitor. And the objective is to see pathological complete responses in ER-positive, HER2-negative disease, which are very rare with chemotherapy. So we start to seeing these dramatic responses, we know we're on the right track. Of course, the problem is for this trial design, you're only offering the experimental drug to about 40% of the patients. What about the other 60%? And here we've been focusing on this target here, CDK4/6. And the reason for this is, interestingly, the PIK3CA mutant population tend to have fewer amplifications of cyclin D1. The wild-type populations more likely to have cyclin D1 amplification, and that's what's being regulated. That's a dominant regulator of CDK4, and so the pathway for the CDK4 inhibitor may be more active in this PIK3CA wild-type population. And the CDK4 inhibitor is showing a nice result in the metastatic setting. Looks very similar to the Bolero-2 result with a rapamycin analog, but you would suspect that you're not targeting the same population here. And so now the trial can offer the CDK4/6 inhibitor to patients who are PIK3CA mutation absent. And the final point from the sort of molecular therapeutics perspective I want to make with respect to specific drugs is the evolving issue of MDM2 inhibitors. Now, luminal-type breast cancer is a standout exception in common solid malignancies because the majority of them are actually P53 wild-type. Although when they're P53 mutant, they are tending to be luminal B and poor outcome. But there's another mechanism to inactivate P53, which occurs, according to TCGA routes, in another third. So about a third are P53 mutant, but another third are MDM2 amplified. And that's very interesting because MDM2 is a negative regulator of P53. And if you inhibit the MDM2 using a nice drug developed by Xiaoming Wang at the University of Michigan, you can actually raise P53 levels in the cell, induce cell cycle arrest, and even cell death. And I thought that would be a very cool treatment for luminal B tumors that are MDM2 activated and P53 wild type. Now, of course, any one tumor may have more than one of these lesions. And so, obviously, we're entering to a sea of sort of therapeutic complexity here that will take probably a decade to unravel. But we do have some very nice drugs to start the journey, as I've tried to point out. And we summarized, I think, the current state of the field with Chuck here in cancer discovery, and I'd encourage you, if you're interested in this, to have a look at that publication. But I think we do have to think very carefully about our diagnostics here. It turning out that the HER2 mutation trial is tricky. Because right now, the sequencing is being done by Sanger, and that basically requires a lot of tumor material, a lot of failures. In fact, I think with Sanger-based sequencing of metastatic samples, you might have as many as 1/3 of the sample failing a Sanger sequencing assay. So, of course, next-gen sequencing is the answer to that, but it's expensive. And, of course, whole genome sequencing, which generate these beautiful pictures of the comprehensive genome of a tumor, costs about $20,000 right now. But, of course, the cost is coming down. So it's quite possible that within maybe 4 to 5 years, your pathology report will include a figure that looks like this. It's very sobering looking at this particular genome, isn't it? So many changes, copy number changes, somatic mutations, and these very interesting crisscross events between chromosomes, which probably result from a sort of catastrophic chromosomal breakage during a cell cycle and then repair. And you can see at the end of these, you've got all these amplification events, and I think that it may even be that these chromothripsis events may be the initiating event in many breast cancers. So I'll end with my compliment slide. I work with very large numbers of people. I've just emphasized perhaps people who've touched on the story I've given you, but in general, thanks very much to everybody for your support over the years. Cheers.
Thank you very much, Matt, for this